Hello, and thank you for joining me for Bruker's second installment of our Lightsheet webinar series. My name is Dane Maxfield, and I'm a Sales and Applications Specialist for Luxendo. In today's webinar, I'm going to focus on telling you about how Lightsheet fluorescence microscopy can be used for your day-to-day live cell imaging and why your next confocal should actually be a Lightsheet. Before I get going on the presentation, I want to point people to the chat window at below on the, GoToMeeting screen. You can type any questions you have in that chat window, and I will try to address them at the end. If your questions pertain to a particular slide, there should be slide numbers in the lower right-hand corner, and you can reference those, and we can head back and look at those as needed. So to begin, I would like to actually get onto this screen so I can change. There we go. I would like to start with this figure I like from the Gold Lab that really shows the different scales at which nature presents itself. And what we can see from this is what I like to show is that with your naked eye, we can see things over a couple orders of magnitude, from things as large as the largest animal on Earth, the blue whale, down to about the size of a hair. And this was the limit of our resolution until the first microscope came around in the late 1500s. And this allowed us to extend our resolution and actually peer inside of cells and be able to observe subcellular structures. And again, this was the limit until the 1930s, when the advent of the electron microscope allowed us to extend this resolution down to the nanometer and sub-nanometer scale. And these have been really important tools for scientists in general, and particularly light microscopy has undergone a couple big transitions in the last 10 to 15 years. There's been the so-called race to the bottom, that I like to say, where you've got the co-development of a lot of these extended resolution and super resolution techniques, such as SPIM, STED, and single molecule localization. And about the same time, there was another push in order to image things in a more physiologically relevant context, such as inside whole organisms or in 3D cell cultures. And in this push, we saw improvements in laser scanning technology, really the peak of spinning disk confocal, and the rebirth of light sheet microscopy. And in particular, light microscopy has been very important and a very key instrument for scientists to be able to study live cells because of the non-invasive nature of light. A popular way to do this is with fluorescent molecules, and these tend to come in two different flavors. We've got organic dyes that can be coupled to antibodies, such as Alexa 647. We'll couple that to an antibody and use that to localize it to a protein of interest. We've also got dyes that can be added to our sampler to the media, such as mitotracker that's uptaken by mitochondria, SIR that binds actin, or Flow4 that reads out calcium levels. And the other flavor of these are the genetically encoded proteins, such as GFP, YFP, mCherry, that we can splice into the DNA of our protein of interest, and as it's transcribed, it will make a Fluor4 for us. All of these function in the same way, and let me find my mouse here, in which they absorb a specific wavelength of light. They'll get kicked into this high energy state, and as they relax and fall back to the ground state, they emit a photon of a different wavelength of light. This is referred to as the Stokes shift. And as we can see on the bottom panel on the right, we see that emission spectrum or excitation and emission spectrum for YFP, and we see that YFP is preferentially excited at 515 and emits around 545. And these emitted photons that we get from these fluorophores are the most prized possession for any light microscopist. Particularly, because all of your fluorophores have a limited number of photons to give. And every time we excite them to visualize whatever protein of interest we've tagged, we're consuming these fluorophores and taking photons away from our budget. So we might be able to visualize this once and get the emitted photons, but as we do this repetitively, what we're going to end up seeing is a decay in the fluorescence signal. And this is because some of these fluorophores end up transitioning from this high energy excited state into the quote forbidden triplet state, where these fluorophores interact with other molecules and become irreversibly covalently modified, also known as photobleaching. Not only does this consume the fluorophore and essentially remove fluorescence from the signal that we're able to detect, but this can also generate reactive oxygen species which can stress and or kill your cells. So the question for everyone when you're doing a light microscopy experiment, and the most important thing to think about, is how do you make the most of this limited photon budget? So every time you start your experiment, before you even put your sample on the microscope, we've already got a set number of photons that we can get from this sample. And that set number of photons is the product of approximately four things. So A, the number of fluorophores you have in there, which is difficult to change, but it's also a product of the resolution that you choose to image at, the number of optical sections you take, the excitation power that you put down in your sample, and the amount of time that you want to image. So we have to budget these appropriately in order to perform the experiment that we want to do. And instead of taking the prettiest picture that we can right at the experiment, we'll have to modify the experimental parameters if we want to image over time by reducing the excitation power, possibly taking fewer Z steps, or taking fewer time points in order to stretch that photon budget farther. However, if we do this, and we only take a beginning time point and an end time point, what we'll see, why did this change? There we go. What we see is that we've missed a lot of development that's going on. And we see in this embryo that there was some pretty massive changes. And maybe we had enough photon budget to squeeze through and get a couple other time points in there. But that really doesn't tell us the whole story of what's actually happening. And in order to get the whole story, we're going to have to actually change how we image. And by that, I mean we need to use a technique that makes better use of the set number of photons, or in other words, we should start looking at light sheet microscopy. But what is it about light sheet microscopy that allows us to stretch our photon budget further? To better understand this, let's first consider the drawbacks of standard imaging techniques such as confocal laser scanning and spinning disk confocal. In laser scanning confocal, if we want to image this particular Z-plane in this Drosophila embryo, what we're going to do is deliver a cone of light that's focused onto that focal plane from an objective, and then raster scan this across our sample, building our image point by point and detecting the emission point by point. In doing so, we've exposed our entire embryo and our entire sample to light, even though we've rejected the vast majority of that with a pinhole and are only collecting that focal volume right around that focal plane. In spinning disk confocal, we can parallelize that a little bit more and get a little bit more speed. So imaging the same plane, we'll use a disk with static pinholes on it, parallelize the excitation with these pinholes, and also parallelize the emission by using an area detector like a camera. But again, it comes with the same drawbacks as confocal laser scanning, whereas we excite the entire embryo with light in order to collect that very specific focal volume that's rejected by the disk of pinholes. Conversely, in light sheet, what we're going to do is we're going to decouple our excitation and our emission pathways such that When we excite the fluor, we're going to excite orthogonally to how we're detecting. That is, we're going to come in with a different objective, deliver our excitation light only across that focal volume, and then from a perpendicular axis, we'll detect the emission profile or the emission light from an area detector, allowing us to capture the entire optical section at once. And this has some key advantages over confocal microscopy. Not only do we have a dramatically reduced light dose, which means that we have less phototoxicity and less photobleaching. In fact, it's estimated that a laser scanning and a spinning disk confocal will expose your sample to 1,000 to 5,000 more photons than in light sheet. Additionally, we have no pinhole to throw away the photons. That is, in traditional techniques where you have one objective to deliver your excitation light and to collect your emission light, we're exciting all of these fluorophores inside the sample and using a pinhole to reject away all the light that's not in the focal plane. Whereas in light sheet, we're only exciting the fluorophores in the focal plane of interest. This leads to a much faster acquisition time, and we can do this with the same 3D optical resolution that we get in these other confocal techniques because of the fact that we're constraining our excitation light axially. This means that this is an ideal imaging technique for looking at dynamic processes in live specimens. And we can turn those static time points that I showed you in the earlier slide into these extremely dynamic movies. And here what we're looking at is a Drosophila undergoing embryogenesis, and we're going to image this embryo every 30 seconds for 15 hours, and then every 10 minutes for 5 hours once all the cool stuff has ended up happening. So we're just watching ventral furrow formation. And the reason that we can do this imaging for so long is our excitation light is no longer rate limiting. Because we're exciting orthogonally to our detection axis, we're putting down a minimal amount of excitation light, which allows us to image for days on end. And I'm going to cut this movie short just to keep going. All right. So now that I've shown you how light sheet differs from your standard confocal techniques, let's take a couple minutes to really touch on some points and highlight these with some experimental and theoretical data. So light sheet has an extremely low photobleaching rate, very low phototoxicity, and high acquisition speed. So why is that? So let's first touch on photobleaching. To give you an idea of the photobleaching in comparison to techniques like spinning disk and point scanning confocal, we've set up a quote-unquote head-to-head photobleaching comparison. And in this, what we're going to do is essentially, it's a busy slide, I understand that, but what we're trying to set up here is that we have are putting these three different systems in essentially the same optical configuration so that we can test the photobleaching theoretically. We've got some references here over on the right and parameters for the equations on the left, and I'm not going to show you the math, but I will show you a couple graphs after it's been plugged in. So the first thing that we're going to look at is the photobleaching rates for imaging a single focal plane at 100 frames a second. We normalize the illumination intensity between these different techniques to be set to achieve or to get 5 photons per fluorophore per frame. And when we do this, imaging at 100 frames a second for 200 frames, we see that in laser scanning confocal, we've essentially lost about 40% of our signal by the end of the experiment. In spinning disk, it does significantly better, where we're sitting at about 80% of our original signal still there, and light sheet is still the best with only about 10% photobleaching. However, these techniques are mainly used as 3D optical sectioning techniques and not for imaging single focal planes. So let's look what happens when we take a stack. When we take a stack, there's a very dramatic change in these photobleaching curves. And this is particularly because in spinning disk and laser scanning, we're exciting all of these fluorophores multiple times in order to get a very small Z series. In laser scanning confocal, at the end of 60 frames, we've essentially consumed every single fluorophore in the system, and we've got nothing left for the experiment by just taking a couple 40 micron stacks. Spinning disk will limp towards the end, but by the end of our 200 frame experiment, we've got essentially no fluorescence left. Compare that to light sheet, where we've lost about 20% of our fluorescence by the end of the experiment, meaning that we can still keep going for a lot longer. And this theoretical data fits in very good agreement with some experimental data from Ernst Stelzer's lab in this 2008 paper, where they imaged yeast with light sheet and with a wide field delta vision microscope. And what they see with this is in agreement with the theoretical data I just presented, is that the light sheet definitely outlasts the wide field technique due to its inherent optical sectioning. So now that we've touched on photobleaching, I next want to focus on the phototoxicity. And instead of showing you, embryos that are resting, that even an armchair biologist can tell you that that's phototoxicity, I want to start by showing you that some of these phototoxic effects are not as dramatic as you might think, and some of them are far more subtle. For example, I'm going to focus on this paper from Biophotonics in 2013, where we're looking at craniofacial bone development in zebrafish. And in A over here, if I can find my mouse, we'll see that from 3 to 4 days post-fertilization, this bone, this operal, undergoes a pretty dramatic change where not only the bone elongates, but the distal end of this bone actually fans out to create the final bone. And what the authors noticed is when they were trying to look at this with a traditional technique, in particular spinning disk, and image this development over time, they saw indeed that when they looked in 3 different bones that had been imaged every 10 minutes for this one day, that they didn't see the same fanning behavior as they did in E, where this fish had only been imaged at 96 hours. They compared that to their light sheet microscope, highlighted up here in B, where all 3 of these bones exhibited the proper fanning behavior after being imaged every 10 minutes for 24 hours, and this matched in conjunction with the single bones that had been imaged either in spinning disk or light sheet at one time point in general. And they go on further to quantify this. In A, we'll actually plot the smallest perimeter versus the largest perimeter to show how this bone is elongating, and we see that highlighted by the blue line in B, blue line in the right, whereas in B, in spinning disk, we see that the bone is still elongating, but it's no longer fanning at the distal end. And so what this really tells us is, although the embryo kept developing, the phototoxic effects that were induced by imaging through spinning disk confocal could have led to artificial results. Whereas the fish imaged with light sheet, we can watch this fanning behavior and do so in a gentle enough manner that it no longer perturbs the development that would have occurred normally. And last but not least, I want to highlight the high acquisition speed of this. And to show you the high acquisition speed of this technique, I want to show you some data from a recent paper from Jan Ellenberg's lab, the EMBL, that was acquired with one of our InVi SPIMs. And here what we're looking at are the very beginning moments of fertilization of a mouse embryo. I'm going to show you the static data first in the movie because it's easier to see with the static data. In phase 1 here, we'll see that we have our haploid sperm marked in cyan, and our haploid egg as they first come together. The cyan is marking centromeres with the minor end, and the magenta, the centromeres with the major end, allowing us to differentiate between the paternal and maternal genomes. And as we see in phase 2, we get the nuclear envelope breakdown, we get mixing of these genomes, and as the cell starts to undergo mitosis, we'll see these line up, and then what we end up seeing is something that had been postulated but never actually observed, is we watch as the cell undergoes division. It undergoes its very first mitotic division. It still segregates into the daughter cells that contain equal amounts of paternal and maternal genetic material, but this material stays spatially segregated as they undergo this first mitotic division. And here I'm going to show you a really blown up movie, and it looks a little blurry, but it's blown up way bigger than it should be, showing you when the eggs meet, they're mixing their genomes, and as they pull apart, we'll see that the magenta and the cyan stay spatially segregated even though they've gone into each daughter cell. And the only way that we are able to acquire this data is by using the high temporal speed and low phototoxicity of a light sheet microscope. So to recap so far, I've shown you the advantages of light sheet fluorescence microscopy, and by changing how we illuminate our sample, we can make better use of our photons, minimize photobleaching and phototoxicity, and still maintain a high acquisition speed. Stereotypically, this is thought to be a technique that is only for, quote, large organisms like zebrafish or Drosophila embryos. And our goal was to adapt that away from this whole organism into a manner that can be able to image cell monolayers, 3D cell cultures, and be able to do this with a high enough resolution to be compared to confocal microscopy. And to do that, we needed a geometry that was really compatible with cellular studies, and thus we created the InVi microscope. The InVi microscope is an inverted light sheet, and I'm going to go into this in a lot more detail in the next couple slides. But here what we're showing is a single illumination lens here and a single detection lens, and our sample sitting between these. And we really designed this with a couple things in mind, which is very simple sample mounting, a very high resolution light sheet microscope, and it has to have the ability to have full environmental control. And I'm going to touch on these in the following slides. So to begin, let's go through the components of the InVi. We first have the incubation chamber that's going to pop up there. And we see that it has two scientific CMOS cameras. Again, as I mentioned before, this is a single-sided light sheet. We have a single illumination lens and a single detection lens. And we are going to use these two scientific CMOS cameras in order to spectrally separate two different channels, allowing us to perform two-color simultaneous imaging. Let's focus a little more on the light sheet generation and the lenses themselves. So here we'll see the incubation chamber and the specimen carrier. This I'll go into a lot more detail in the next slide. We see our illumination lens, which is a 10x 0.3 NA, and our detection lens, which is a 25x 1.1. Now 25x might not seem as such magnification that you might want, but because of the high numerical aperture of this lens, we can post-magnify this with tube lenses inside the system to get final magnifications of 31 or 63x. And I also want to point out this is an extremely high-speed imaging system. So for each camera at full frame, we can run more than 75 frames a second. And that's per camera per color. So for doing a two-color simultaneous acquisition, we can get closer to 160 frames a second by taking both of those colors at the same time. The specimen carrier is really the crux of the InVi, and this is what makes this thing work. So here's a side view of our illumination chamber, or of our incubation chamber. We see that we have a single illumination objective, our single 25x 1.1 Na detection objective, and both of these objectives are water immersion objectives. And we use water immersion objectives for their optimal properties of long working distance and high numerical aperture. Now, because your cells aren't going to like being immersed in water, what we use is our special sample chamber that has an FEP foil barrier that creates a physical barrier between the water environment and the cells. Now, FEP has a similar refractive index as water, which essentially makes this an optically clear barrier between our imaging objectives and the sample. This also allows us to fill this chamber with whatever sort of media that your cells want to be in, and either culture your cells in these chambers or physically place them in, and we can line up multiple of these embryos at a time to perform our imaging. And then generate a light sheet with our illumination objective and detect the emission orthogonally with our detection objective. Couple quick specs on the light sheet. This is a digitally scanned light sheet, so our light sheet is created with a Gaussian beam that we're going to digitally scan to create the sheet. We have an adjustable beam waist thickness, so we can adjust the optical section thickness for our imaging, and we also have line mode for improved background suppression. So since light sheet is so gentle and the imaging is not affecting the specimen's survival anymore, there's one more issue that becomes important, which is environmental control. And here what I'm showing you is some of the components of our InVi SPIM incubation chamber. You'll see that we have temperature control. We actually heat the entire incubation chamber, allowing us for very precise temperature control of the media that the sample's in. We also have a heated lid to make sure that the whole chamber stays hot. CO2 and CO2 and O2 control, which allows us to create the environmental conditions that your cells want to be in. And additionally, we can perform hypoxic and hyperoxic conditions in order to study your cells. And last but not least, humidity control, so our sample and media is still there and hasn't evaporated by the time we get done with our long-term experiment. And with all this in place, we can image seamlessly for days on end. And here what I'm showing you on the left is a tumor, and we're watching this grow by actually switching on the expression of an oncogene with deoxycycline. And we're going to image this tumor growth for 2 days. And on the right, what I'm showing you is that exact same tumor 2 days later, after we've washed out the deoxycycline, turned off this oncogene, and now we're watching this tumor undergo apoptosis. And in fact, the blue cell here, or the blue dot marking the cell, is one of these cells that essentially escaped death and is moving out from the inside of the tumor to the outside. To give you an idea of some of the other samples that we can image with the InVi SPIM, we can accommodate a very wide range of samples. So here I'm showing you some microalgae on the smaller end of spectrum. We've got a section of mouse cochlea and a zebrafish pituitary gland. So both of those can be easily inserted into the chamber. In a Arabidopsis root and a zebrafish embryo, just the dorsal view of this. And last but not least is one of my favorite experiments, watching mouse oocyte development. And so here, what this experiment is showing is the high temporal resolution of the light sheet and the low phototoxicity. We're watching this mouse embryo develop from the two-cell stage, and we're going to watch this development for 3 days until it hits the 63-cell stage. These individual nuclei or cells can be tracked based on their cell lineage. And so what you'll see is the little balls that are marking the cells will actually change color as the cells determine their fate, either into one lineage or the other. And it's only because of the high spatial and temporal resolution and the low phototoxicity of the InVi SPIM that we're able to capture these movies for such a long period of time. And the one part of this that isn't in this experiment, but is extremely crucial for this, is that after we've imaged these embryos for 3 days, we were able to reimplant these into a pseudo-pregnant female mouse, and the baby mouse that came from this developed normally. And this really highlights the gentleness at which this sample was handled. Otherwise, these would arrest almost immediately. And for those of you that have different samples, I want to give a quick plug for some of our other products out there. I've already told you about the center one, the InVi SPIM, and about how that is a high resolution light sheet. I also want to give a plug for our movie SPIM, which is a horizontal geometry composed of 4 objectives. This is really built around imaging larger specimens such as Drosophila embryos, zebrafish, and even cleared tissue. And on the right, we have our upright geometry, the QV SPIM. This has matched objectives and can accommodate isotropic resolution of things that you want to image. And the nice thing about these is they're all kind of all built off the same platform. So we have one platform that we create 3 different light sheets from. And our literal black box, it's a gray box on here, is what contains all the embedded control electronics which control the experiment. There's no external control hardware. We also have our laser hub that you can configure how you'd like, adding up to 6 lasers, whatever your experiments need. We provide a high-performance PC in order to control the acquisition and for some local storage. We have an optional storage and processing solution, and we have our dedicated light sheet control software. And with this infrastructure in place, we can then pick what light sheet geometry that you'd like, whether you want the horizontal 4-objective multi-view SPIM, the inverted InVi SPIM, or the upright QV SPIM. And with that, I'd like to take a couple minutes to go over our take-home messages for this webinar. I hope by the end of this that I've showed you that light sheet fluorescence microscopy is suitable for your everyday imaging. We have a wide variety of sample mounting techniques with our different light sheets that can accommodate your experimental needs. And the real advantage of light sheet is performing your live cell imaging, allowing for very fast volumetric acquisition with very low phototoxicity and photobleaching, and we can achieve very high resolution at large fields of view. And for the vast majority of these systems, we also have a photomanipulation add-on that can be added in order to perform FRAP, photoconversion, optogenetics, or cell ablation. And with that, I would like to end. Like to thank everyone for your attention and for either waking up early or for taking time out of your day to see this. My contact information is below, and I can take any questions that you might have. Thank you. Okay, we have one here. What's the difference between the movie and the InVi, and can I put both on one system? The difference is in the sample mounting. Let me go back to this. One more slide. Here we go. Okay, so the difference between these is how we're actually mounting the sample. So you'll see in the InVi we have this very specialized sample chamber that we can grow our cells on the FEP foil. On the movie SPIM, we're actually going to mount samples either in, for live cells, either embedded in agarose in a capillary or in an FEP tube, and place them between the 4 objectives. Or for cleared tissue, we have different mounting schemes in a different chamber. And we've got one more question here. How difficult is it to prepare the sample for the InVi SPIM? It actually isn't. It depends on your sample. So for cells, because the FEP is a little bit hydrophobic, we tend to coat this with some collagen or polylysine. And once we do this, they'll lay down like they would on any glass surface. For organoids, embryos, and spheroids, this actually is an extremely easy way of mounting it because we can just physically drop them into the chamber, line them up, maybe 10 or 20 depending on the size, and then serially just image each one of these individually to get us a real high throughput way of performing our imaging. And last one, why do you have 2 cameras on the InVi? The 2 cameras are for, let me get to this, are for simultaneous 2-color acquisition. So although we only have a single-sided light sheet, we can spectrally separate the channels by turning on a red and green laser, and then just have this camera see green and this camera see red, allowing you to look at 2 colors simultaneously and thus speed up your temporal resolution. Okay, I don't see any more coming in, but if anyone else has additional questions, my contact information is up below, so feel free to reach out individually, and look forward to hearing from you. And thank you again for attending the webinar.